Let's start. It is a great pleasure to welcome you all to this symposium. It is the 10th CNAG virtual symposium on genome research, de novo sequencing and assembly for biogenome projects. I want to invite you here and say hello to you in the name of myself and the CNAG and my co-organizers. And what I'll do now is I will switch over and show you my slides, if I can find them. This is here. Okay, so let's go. I want to welcome you all to this symposium. It's a delight that you're here. We are co-organizing this event together with ERGA, the European Reference Genome Atlas project, and the CBP, which is the Catalan BioGenome Project. It is the 10th time we're having such a symposium, but it's the first time that we do this virtually due to the circumstances. We don't have to go into that. I'm going to quickly run over a few things that are of importance. The first is housekeeping. I want to ask all speakers to please stay on time. Make sure that you don't run over. We have a very tight program. In contrast to when we do this symposium in person, we have shortened the talks and we have shortened the entire procedure. So we have usually a one-day meeting. Now we've sort of compressed that into 3.5 hours, simply because we know out of experience that sitting in front of a computer for many, many hours is a little bit taxing. On the other hand, we make the talks shorter this way. We get more packed in. Same number of talks, shorter talks, but more condensed. We want time to answer questions, the participants' questions. That means the speakers have to really stay to their allotted time. In order to ask questions, please type them into the Q&A. In the Q&A box, you can then vote for questions that you see. You can say, that's a question I'm also interested in. Then you label it and it goes up to the top. And afterwards, when we then get around to posing the questions to the speakers, I will pick the ones that are on top. I have a little committee here made up of me who's going to take a decision on which question is going to be asked. And then we will go through that. And then we move on to the next speaker. We will not be using the raise hands function, so you can put your hands up, but we will not be looking at activating people to ask the question themselves. We will do it through the Q&A function in order to sort of keep the flow of it. Feel free to use the chat, but beware who you send something to. So if you send it to someone directly, beware, or if you want to say something naughty to someone about someone else, be very cautious to send it to the right person, not to the wrong person. This would be not very nice, and obviously it's the responsibility of the users of the chat and not our responsibility. We don't edit anything on that side. Good. Okay, so we have an organizing committee for this. This meeting. It is made up of Ana Riesco from the CSIC National Museum of Natural Sciences, Montse Coraminas from the UB Faculty of Biology, and Rosa Fernandez from the IBE here in Barcelona. So I want to start out by thanking them for helping us with the organization of this, picking the speakers and all of this. Okay, so what I'm going to do first is give you a little bit of an overview of CNAG and what we actually do. So just to say what our mission is that we are meant to carry out large-scale projects in genome analysis that will lead to significant improvements in people's health and quality of life. And obviously, biogenome projects, the environment is a very important factor in the quality of our lives. So we do this with local researchers, Spanish, European, international research community, but also with the clinical community and also with industry. As a center, we view that we want to be really good at what we do. So we want to be a world reference in genome analysis. a quick skip over this. Marta is going to show more about this in a minute. We have a whole bunch of sequencers, and we have a whole bunch of computers, and we put these two things together to do our work. So we do sequence analysis and we do interpretation of the data that we generate on our sequences. We work around a process. The process is tracked by a laboratory information management system. We go all the way from study design, where a project is input into a system through receptioning samples, doing the lab work, doing all the preparation, doing the sequencing, quality controlling the data, doing analysis, and transferring data plus results back to our collaborators. This is how we work every project. And obviously, many projects cover the laboratory part as much as they cover the analytical part out the back. And there's a lot of bits and pieces that go into doing this well. Just a word on the applications. We obviously run different applications. We do DNA analysis. We go all the way from whole genome sequencing to refined targeted sequencing protocols such as GPS. In the middle of this, obviously, capture technologies to pull out the exome, or to pull out panels that we have an interest in. We can do this with low amounts of DNA and out of very challenging materials such as FFPE or DNA extracted from circulation in humans. RNA, a whole series of protocols as well. We have protocols that go towards the Illumina sequencers, protocols that go towards the Oxford Nanopore Technology sequencers. We can directly measure RNA molecules, and we can do this again at different levels. We can do it in bulk or all the way down to single cells. And then in epigenetic analysis, we have DNA methylation analysis in different forms, whole genome bisulfite sequencing, OxBS. We can do this with low input. We have single cell attack. We have attack in general, and we also have high-C that can be done in bulk. Then our informatics resources, a whole series of different teams and groups and units that have different levels and different kinds of expertise, all the way from production bioinformatics, where we do all the clerical work, tracking, making sure that what data we have, to analysis platforms that take care of DNA protocols or RNA protocols or integrative analyses and so on, to de novo assembly and annotation that you will see in a minute as well. We develop algorithms, we do modeling of the nucleus, we compare genomes to each other, we do this in the context of biomedical, we do it in the context of population, and we build databases that are very popular. Example, given the ArtiConnect genome-phenome analysis platform, but that's not the topic for today. Very important also is that we have a quality system running. The entire center runs under ISO 9001, and we have ISO 17025 accreditation for certain ones of our processes, particularly the ones that are closely related to clinical work. We are Illumina certified service provider, Roche certified service provider, Oxford Nanopore Technologies certified provider, and we are also a BBMRI ERIC expert center. Okay, and with that, I'm gonna sort of hand off the procedure to my co-organizers. I'm gonna ask Ana Riesgo to say a few things about the ERGA. Okay, I'm gonna stop sharing at this point. Okay.
Thank you. Thank you, Ivo. Thanks for giving me the chance to present here this European initiative. Can you see my screen?
Yes.
Okay, good. So as Ivo said, I'm Ana Riesgo from the National Museum of Natural Sciences here in Spain. And I'm going to talk to you about this European reference Genome Atlas project for sequencing European biodiversity. So we are a pan-European scientific consortium of people to give a response to current threats to biodiversity, basically because 1/5 of the 200,000 European species are at risk of extinction. And we need to give a response to this, and this response, of course, should be orientated to protecting species and ecosystems. But for this, we need to understand biodiversity. There's many ways of understanding biodiversity, but one of them is by studying genomes of the species. So our proposal is to establish an interdisciplinary workflow to obtain high quality reference genomes to aid protection of European species. So we plan to work from species to genomes and beyond, of course, to understand these genomes and the adaptation of these species using these genomic resources by involving museums, sequencing and computing centers, and of course, research institutions. So our aims are basically creating and consolidating a collaborative and interdisciplinary network of scientists across Europe and of course associated countries, connecting relevant infrastructures across Europe following a distributed model that can dynamically increase, of course, and then propagating guidelines for state-of-the-art genome establishment through training and knowledge transfer. Who are we? We are now hundreds of scientists across the entire Europe, but it's basically an initiative or a consortium that's formed with a bottom-up approach, and the whole idea is based on people. So to gather all this expertise and knowledge about biodiversity genomics, but of course involving research institutions and infrastructure facilities such as CNAG, but also partner genome projects. So we build on the expertise and the capacities of the Earth BioGenome Project. And we have regional partners such as the Darwin Tree of Life, but of course the Catalan BioGenome Project that Montse will introduce in a minute, and this endemic seed, of course. And then worldwide partners such as the Vertebrate Genome Project and other large-scale projects for biodiversity genomics. So this is a consortium to respond to these current threats, and we will we will start doing all these biodiversity genomics in the different centers, research centers and sequencing centers. But here in Spain, we already started with a pilot study involving CNAG and other research institutions to sequence the genomes of these 4 species that are important for different reasons in the Iberian Peninsula: sponge, Fakelia ventilabrum, spider, Macrocele calpellana, this clam that is Venerupis corrugata, and this worm called Gordius gonzalezi. So we have already started them, and we plan to sequence them with Promethean, with Nanopore, and with NovaSeq Illumina platforms. This is not a closed project or a closed initiative. We welcome all people. So if you are interested in this initiative, you can join us here in this website. And that was all. Thank you very much.
Okay, thank you very, very much, Anna. Okay, now we will move over to Montse, second co-organizer of the meeting. So Montse, can you please come in and show your slides?
Yes. Can you see it, Jason?
Yes, perfect.
Okay. So thank you. I would like to briefly introduce the Catalan initiative for the Earth BioGenome Project of the CBP, which aims to sequence all eukaryotic species in the Catalan territories. This initiative was initially driven by the Institute of Catalan Studies and 2 of its societies, but as it's mostly funded by the Institute of Catalan Studies. But we have now more than 20 official partners of institutes and research centers, and close to 100 members. We are an affiliated project of the Earth BioGenome Project, which means that we are a node for this international network of networks. And as Anna said, we are also a partner for the Earth, right? We are one of these regional partners, as you can see at the bottom. Why we want to do that? Because we think that this region is a biodiversity hotspot, because there is a rich and long naturalistic tradition and also strong research centers in biodiversities that come from universities and many other research centers, museums, etc. And because we have the leading sequencing and computational infrastructures. I just would like to say that we were sequencing currently 31 species, and the funding for sequencing these species comes from a first call that was launched in 2020, and also from own funding from research groups. And I'm very happy to announce that a second call will be launched next week on Thursday. So if you want to know more about this second call, or you want also to contact us and join, you can do all this through our webpage and just biogenome.cat. Thank you very much.
Okay, thank you very much, Monse. Thank you very much, Anna. So, okay, so you see the two initiatives that are sort of bracketing the meeting that we have today. Good. I will continue in the proceedings. There is a question in the chat, Stefan. I will answer you directly. So now it's my great pleasure to move on to the next speaker. The next speaker is Marta Gut here from the CNAG. She heads up the sequencing unit. So Marta, if you can share your screen, that would be nice.
Thank you, Ivo, for the introduction, and thank you for invitation for me to present here at the annual symposium on the genome research. And this year it is on de novo sequencing and assembly that we actually have quite a lot of experience. Since the last 10 years, we have done a few of the genomes, but the laboratory techniques for the de novo sequencing improved since then really a lot. And the de novo sequencing have been using before particular techniques that were essentially compatible with the short read sequencing that we have here. And we are very well equipped on the level of the sequencer, on the level of the quality that we provide to our collaborators, the ISO accredited results. But also at current, still we are using the short read sequencing as an important component of the de novo assembly pipeline. We use the proximity ligation protocol that allows the chromosome scale super scaffolding. So still there is a space for short read sequencing. I term it as a long shot, bringing the long distances by short sequencing. So the long reads provide the information over the repetitive regions that are the structure rearrangements, large insertions and deletions, or segmental duplications that are not possible to interrogate with the short read sequencing. And this allows to assemble the genomes more complete and to extend into the centromeric regions. Pacific Biosciences is one of the leaders in the long read sequencing that is I term as the short long. Their approach to interrogate the long single molecules is similar to the Illumina sequencing by synthesis, where polymerase is synthesizing the complement of the original strand by incorporating the heavily modified fluorescently labeled nucleotides. This sequencing mode that is depicted here is called the continuous long read sequencing, and passing once around the original library, we get the raw read accuracy of about 90%. And that's actually also the reason why PacBio is slowly abandoning this sequencing mode, because the circular consensus sequencing mode results in much higher accuracy. And that was due to the polymerase that is capable of actually holding the single-stranded circular DNA much better and can pass around. And because it has strands displacing activity, it can synthesize a concatemer that is then being in silico deconvoluted. And after more than 10 passes around, we can achieve the accuracy 99.9%. That is really very important achievement in the long read sequencing. And these reads are called the hi-fi reads. PacBio has in the field just one sequencer, whether in the iteration of sequel 2 or sequel 2E. And both of them, they are able to produce the hi-fi reads on the serially process up to 8 smart cells. The hi-fi reads have one or actually two limiting factors. Firstly is the length, because the single-stranded circle DNA is not easy to hold on by the polymerase. The length read is limiting usually to 15 to 20 kilobases. And the second limiting factor is actually the yield. If one of these flow cells is capable to produce 50 to 100 gigabases, only fraction of it are the high-fi rates. In Oxford Nanopore Technologies, the yield and the length is certainly not the issue, but there are other limiting factors I will be speaking about. But it is a technology that we have here in the CNAG, and we have quite a good grasp on it. We are a certified service provider, and annually for that reason, we are evaluated for providing the state-of-the-art quality to our collaborators. There are 4 different sequencers from ONT. They vary in portability, in yield, size, and also computational autonomy. The most powerful is the PromethION. That is actually the most powerful sequencer at the moment at the market. And it comes in 2 formats, whether to run 24 or 48 flow cells. And these flow cells can be started and finished anytime in function of the protocol or in the need of the user, which is the same for the GridION. As in the CNAG, we all have all the 3 instruments, the PromethION, GridION, and the MinION, but obviously, mostly we are using only the 2. The Nanopore technology is depicted here. What are the bases where the protein nanopore is being set in an electrically resistant polymer membrane. And by setting a voltage across this membrane, the ionic current is passing only through the pore. But then also the DNA is passing through that pore. That creates a characteristic disruption of the current, and it's being registered in these squiggles, and those are being analyzed by base calling software. The Oxford Nanopore is at the current use state using 2 different pores, the R9s and R10. In the GridION, MinION flow cells, these are both in the production. In PromethION, there is only the R9, and the R10 is coming. The difference is that in the R9, there is a very sharp reader head within the barrel of the nanopore, and approximately 5 bases dominates the signal. While in the R10, the barrel is longer, and there is a dual reader head that enables improved resolution of the homopolymeric regions, because more bases can dominate the signal. And that also can improve the consensus accuracy of the nanopore sequencing data. So what is the limiting factor if the yield is not the limiting, if it's really the maximum yield from the PromethION flow cell has been lately reported as the 245 gigabases, and the longest read is 4.2 megabases. So if it's not the yield or the read length, it is the raw read accuracy that at current is at 98.3%. However, as in all cutting-edge technologies, the speed of the appearance of the latest improvements is enormous. Only in the last 2 years, and that in the improvement of the base callers, we gain 6% in the raw read accuracy. So today with the GAPI5, we have the raw read accuracy 98.3%, and together with the Q20 Plus kit, we have the accuracy 99.3%, and that is detecting after the one single pass of the read and independent to the read length. Speaking about the read length, I would like to just show what are the next development also in that side. We are using a standard ligation kit in the field samples that we currently processed, with the really respectable results of yield of over 60 gigabases and then 50 over 40 kilobases, with the longest base code rate of 1.42 megabase. But if you compare it to the latest kit, which is called the ultra-long DNA kit, the yield doubles, and the N50 as well. I enjoy looking at this, but as you can notice, this is not at the same scale at this experiment. Bringing it there, you can see that that there is a missing the data, the enormous amount of data about which is longer than 100 or even 200 kilobases. So this kit is already on the market, so it's already available. The disadvantage is that the input material is 40 micrograms of DNA, which can be sometimes actually the entire organism. I have been thinking about the rules that underline the success of the long-read sequencing, whether it is PacBio or Oxford Nanopore. It is in contrast to the original Illumina, actually the purity and the integrity of the DNA that is really, really important. And I have selected few rules that should be thought about already ahead before you start to do the sequencing experiment. All these steps already matter. So if you have your organism, select well the tissue that you will be extracting the DNA from. So rather the ethylated young leaves than the dark green leaves, or in the animals, rather the spleen or kidney rather than the gut or the brain. So, and the material obviously has to be fresh and clean. The freshness is logical because the deteriorated material will have as well deteriorated DNA. But the clean, that can already, the debris can hamper even the DNA extraction protocol. So this is very important that the material, the organ that you are storing is clean. The best way, if you can, is to flash freeze in liquid nitrogen using isopentane. That is, of course, very easy in the laboratory, much difficult in the field. And store the organ or the tissue in -80 or in liquid nitrogen. Do not let the DNA, do not let the sample being exposed to fluctuated temperature. Already at -50, the DNA gets brittled, the crystal starts to appear. People often ask about the DNA extraction method. So regardless the 4 previous points that usually people already have done something to the sample, and then the extraction method should come as the solution. So that often doesn't work so well. But after all, everyone has to understand that each organism has its own best extraction method, and to take into account the primary metabolites, the polysaccharides or polyphenols, or secondary metabolites like toxins or pigments. So all this can influence the quality of the DNA, in particular the purity, but also the things that come from the extraction protocol itself, which can be phenol or alcohol or even high salt content, can be important. So it's important to choose the right method for the right organism, taking into account the particular quality that it has. We are using in a CNAG a lot of the circulomics kits for the extraction, and that is using the nanobind disc where the DNA is being bound. And then after the elution, we also perform the circulomic short-read eliminator that is based on precipitation, the size selection. So with this material, we start to prepare the libraries. And so leave the libraries really for the experts who are in particular in close vicinity of the sequencer. Because unlike this short read sequencing as an Illumina, you can keep for years the library frozen. This is not really anything recommended for a long read libraries, not only because of breaking the long strands, but as well, those are proteins there. So these will really not like the storage and the freezing and defreezing. So here I will finish with the little teaser of the organisms that we have been working with in the past years. And at the end, I would like to thank the entire sequencing unit who is giving great support, infrastructure, and sharing a lot of different protocols with the long read team that is important for implementation and improvements of the protocols. In particular, the team I would like to mention is Nuria, Javier, and Cristina, who are giving great support to the stars of the long reads in the CNAG, Ignacio and Paola. Obviously, at the end, I would like to acknowledge our stakeholders and you for your attention.
There haven't been any questions in the Q&A. I'm sure some of them will appear in the overtime. We're running a couple of minutes late, so if that's okay with you, I will move on to the next presentation. The next presentation is by Tyler Aliotto, who is the team leader for the de novo assembly and annotation team here at Synagogue. So, Tyler, can you please share your screen?
Okay, is everything good? Can you hear me and see me and see the screen? Great. Okay, thanks for the introduction. I lead the assembly and annotation team, and I'd like to acknowledge my team members, Fernando Cruz and Jessica Gomez, two bioinformaticians that help with running the assemblies and annotations and improving the methods and pipelining them and everything. Just a quick overview of what my team does in kind of schematic form. Here I'm going to hide my video. There we go. Basically, we do the assembly, so it involves detecting overlaps between the long reads that Marta was talking about, deriving the consensus, and then we do the structural annotation of genes and also the functional annotation. Okay, in this talk, I'm just going to talk about the first part, doing the assembly. And we look forward to a talk a little bit later from Roderick on annotation. And by the way, it's from Roderick's group that it's where I did my postdoc work, and then came to the CNAG to do both annotation and assembly. The ultimate goal, of course, is to generate complete finished genomes. Okay. With bacteria, we're already there. We can generate circular genomes of high consensus quality. We're doing that just fine. And if you've read the news recently, we can do this for the human genome as well. The telomere to telomere consortium has now generated a complete assembly without the 5 gaps that I'm showing here, using a combination of PacBio hi-fi reads and Nanopore ultra long reads that Marta just talked about. So to have the complete genome, we need to throw many resources and quite a lot of and plenty of manual curation of the genome. So that involves a lot of resources, sequencing and human resources. So the current objective for the Earth BioGenome Project and ERGA and CBP is to arrive at, to achieve genomes that meet this minimum quality, which is actually really, really high quality genomes. not necessarily complete, but 1 megabase contig N50, chromosome scale scaffolds, low error rate, at least QV40, which is 1 error in 10,000 bases, and some additional requirements to have, few duplications, high completeness measured by either k-mers or by transcripts or by BUSCOs, okay, and have a large proportion of the sequence assigned to chromosomes. And ideally separating out any contaminants. If there's symbionts, the organelle genomes, nicely separated and annotated. And if possible, the alternative, let's say you have a primary assembly and the alternative assembly. And this could either be an incomplete set of haplotypic sequences or a complete second haplotype genome assembly. Okay. Over here, I just show a few of the genomes, not all the genomes that we've assembled over the last 10 years, but a select few. Just to give you an idea, they're not, so here they're ordered in terms of contig N50, not necessarily in terms of date. Okay, I thought this was a nice way of listing them. And you can see over the years, we've always used short reads because we're starting out as an Illumina sequencing center, and then introducing long reads. Okay, you'll see that most of the most contiguous genomes are using long reads. You can see the shift that we had from using mate pairs to long reads. Also, there was a period of time that we're using Fosman pools. Okay, and depending on the organism, sometimes we have a genetic map or a physical map. In the case of the fruit fly, it's a physical map. And then various other technologies. And the more recent long-range technologies I'm going to talk about in a minute are 10x genomics linked reads and proximity ligation protocols, HiC. And you see that some of our recent, these happen to be our more recent assemblies. We have a peacock, sole genome will be talked about later on, as well as the linked species that Jose Godoy will be speaking about. And you see that the contig N50s in terms of kilobases are actually in terms of megabases here. 3.6 megabases, 23, 67 megabase, contig N50, meaning no gaps. And then with additional information from HiC or from linked reads, we can boost these contiguities up even higher. And for example, with the peacock, that's chromosome scale. You can't go any further than 93. The last three here listed meet the criteria for the EBP. We're over a megabase contig N50, and we're at or near chromosome scale super scaffolds. And the quality is actually quite high. It's above Q40. So I'd like to talk about the current data types and the methods we use to assemble those. Now, Marta just talked about the data types, so I'm not going to speak about the sequencing, except that we use primarily nanopore sequencing, though on some projects we have also used PacBio CLR reads, okay, for the muscle and for several of the linked species that Jose will talk about, okay. For assembly, we've moved from short read assembly and even hybrid assembly to just long read assembly. So taking just the long reads, detecting the overlaps and constructing assembly graphs, and then essentially detangling the assembly graphs. Okay. Various assemblers have been developed to do this. Canu from Adam Phillippe's group, next de novo, Shasta, and also Fly, which I show a schematic from the Fly paper right here. And we like Fly. The latest versions of Fly are doing very well. They produce very few misassemblies and very high contiguity. We've also in the past been fans of using Mazurka. And Mazurka is essentially now just a preprocessor correcting long reads with short reads, okay? And actually we use Fly to assemble these, what are called super reads into contigs. We're moving away from this approach because we think that fixing the errors after assembly now works better than fixing them before assembly. And that has to do with the quality of long reads that we're getting now. The nanopore reads have fewer errors and they're easier to assemble. So after we have contigs, we like to polish them. Okay, there are some remaining errors. This is kind of an old slide, so it looks like there's more errors than we get now. Okay, so we can do this using the long reads themselves, using just the fast queues in base space. And commonly we use Racon or Medaka, a combination of those. And there's some other polishers now which do work with both long reads and short reads, like Hypo or NextPolish. In the past, we used Nanopolish, which used the raw signal. We've moved away from this because it's more complicated to use, and we're getting just as good, better results with these other methods. And if we have short reads, we still find them useful to boost the quality of the sequence even more. Okay, we have been using Pylon, but like I said, there's some new polishers like Hypo and XPolish which use both, and we're getting good results with those. There's also HaploG, which it does a haplotype aware polishing, and so we're experimenting with that as well. After polishing the genomes, the assemblies, then the one essential step is to remove any false duplications. This is a requirement for VGP, for EBP. It's often the case that we end up with some haplotypes remaining in the assembly. And so right now, the best practice is to use purge dupes from Richard Durbin's group, which uses read coverage. So the reads are mapped to the assembly, and using the read coverage and self-alignments, we can identify duplicated sequences and even detect overlaps at the ends of contigs where they should be joined. So oftentimes we remove the dupes and we end up with a little bit higher contiguity at the end. And so the output is basically the purged assembly and an alternate set of sequences. Then we're ready to do additional scaffolding with other long-range technologies. So even though the long reads are really good, we can use several other techniques to boost this to get to the chromosome scale. One method that we've been using in the last year or so, even though it may be discontinued, is linked reads. Linked reads from 10x Genomics allow us to further scaffold the assembly, kind of to an intermediate scale. Optical mapping is another approach that is being used on some projects, though we don't have the hardware in our lab. Sometimes we have data from on other projects. And optical mapping is a complementary approach. Okay, and this can help achieve chromosome scale. If we have a genetic or physical map, we can use software to, let's say, map the markers to our assembly and figure out the order and orientation of our contigs and put them into linkage groups or physical chromosomes. So for some projects, we have that data, and it's very useful, especially with starting assembly that's already highly contiguous. And then a technique that works with most of the samples, though it's sample dependent, is Hi-C. And this is kind of the go-to method in many of the large-scale projects at the moment, like DGP and Darwin Tree of Life, is to run a Hi-C based scaffolding, okay? The long-range information is used to order and orient contigs into chromosomes. And afterwards, you can see the result. This is a snapshot of the output of dovetail high-rise scaffolding for one of our projects. And you see that we end up with the expected number of chromosomes for this species is 13. And it's great. You can see they're separated by chromosome here. And you see that there's very few contacts between chromosomes. So all these techniques, all these data types, I've put into an overview of all the different workflows that we do and we're working on. So it depends on your input data type. If you have ONT and Illumina, we can run Mazurka or Next de Novo or Fly, Polish. If we have linked reads, we can scaffold with linked reads, right? If we have high C data, we can put these into chromosomes like this. Is this for eukaryotes, and for prokaryotes, we have a separate pipeline here that's much simpler. So if you extract one of those paths, you know, for one particular species that we've done recently, the peacock. In this case, we use ONT data and we had linked reads. We performed a hybrid assembly with Mazurka in this case, where the linked reads were used to kind of correct the long reads. Then they were assembled. Then they were scaffolded with those linked reads. And then we had a RIMA high C. We used both SALSA2 and 3D DNA, two different algorithms for assembling this into chromosomes. And we compared the two approaches and curated the assembly and ended up with nice super scaffolds. For most of these genomes, we have a separate pathway to assemble either mitochondrial genomes or chloroplast genomes. Okay, and this is a nice drawing that my daughter made in the hospital just yesterday for me, just for this presentation. And then this is the second to last slide. I'll be done in just a second. This is our current pipeline that we prefer to use, using long reads from ONT and short reads from Illumina plus high C. So we run ideally Fly, with some cases Next de Novo has given nice results. Polish, several rounds of polishing with both types of data, removing the duplicates and doing the high C scaffolding. And in JuiceBox assembly tools, making additional joins or breaks, kind of correcting the final output and assigning chromosomes. And how do we determine what the best assembly is, or whether we meet the criteria that we've targeted? We run a number of evaluations, including BOOSCO, which is looking for conserved genes. Running Mercury, which calculates k-mer completeness, also gives you an idea of how much duplicated sequence you have, and gives you also the consensus quality, the QV value. And like I said, running JuiceBox assembly tools to fix the contact maps and end up with chromosome scale assemblies. The remaining problems are kind of the same as always. Sometimes we have samples with high heterozygosity, especially in structural variants or presence-absence variation. Sometimes we have samples where individuals were pooled, so we kind of have a population. And sometimes we have highly repetitive genomes that even with the current long reads, we still end up with fragmented assemblies. But these are going to be addressed as the read lengths continue to increase. We get better and better assemblies as the accuracy increases as well. So ultra long kits, the Q20 early access chemistry, and maybe in the future, this kind of Q30 duplex reads that Nanopore is talking about. And some switches in the algorithms used. And this is, we're going to keep pace with the chemistry by switching to assembly algorithms that take the high accuracy into account. So it's very important to match, let's say, the overlap length and tolerated errors correctly to the chemistry. And so we continue benchmarking. And that's all. I'd like to wrap it up and take questions if there's time.
Yes, thank you, Tyler. That's very nice. You do have a couple of questions. I will start with them. So the first one is that Romy Peña does not see how Hi-C adds information to complete de novo assembly. So maybe you can repeat that one.
Yeah, well, I breezed over it for time. So Hi-C is done in situ. So the DNA is not extracted before the cross-linking is performed. So basically, the cross-linking in the cells is done before DNA extraction. So the chromosomes are intact. Okay, so and there is a relationship between in 2 dimensions along a chromosome. So you have more contacts with sequences that are closer together along a chromosome and fewer contacts as the distance increases. So within a chromosome, you can use this information. Okay, and you're basically getting short read sequences next to that are ligated together. So you can tell what's close to what. And these order and orient your contigs into chromosomes. And there are fewer interactions interchromosomally. So you always have more interactions within a chromosome than between chromosomes. Yeah.
Okay, great, thanks. The next question is, how reliably can you assemble regions containing highly polymorphic and highly repetitive genes, such as those in the major histocompatibility complex? This question is by Seraina Emilia Bracamonte.
I don't, that's something that we're working on, okay? And with your group, Ivo, we're also working on that. So how well can you do it? With, I think with the latest accuracy, we're showing that if we rebase call reads, we can start to distinguish types much better. So with the newest base calling algorithms and the longest reads, this should not be a problem. How do you do it? It's, yeah, the chemistry is solving this for us. Okay, essentially.
And then we have one question from Gareth Linsmith that sure. is, it looks like you've tried both Dovetail and ARIMA for Hi-C. Do you have any preference, any comments?
So the ones that we've tried now are the standard Dovetail protocol, which was the Chicago libraries followed by regular Hi-C. And then we tried ARIMA, and now we're testing the OmniC also from Dovetail. Yes. What are our preferences? We had good results with ARIMA. The Dovetail result on one of our species was good, but it was hard to work with the resulting data to curate the chromosomes because of the format that was given to us from Dovetail. So we prefer to do the scaffolding ourselves. We like to have the data and do the scaffolding ourselves. So we prefer to have the kits. Marta's group is doing a great job at producing the libraries and doing the Hi-C. As in terms of ARIMA versus OmniC, we don't have any conclusive results yet. We're in the process of doing those benchmarks.
Yes, thanks a lot, Tyler.
Yeah,
Tyler, you have a couple more questions in the Q&A. Can you please answer those?
Sure, I'll answer them as
I make one comment. This is a big comment about the Hi-C, and that is that actually we had very good experience with the link trees coming off the 10x genomics, and it's actually a shame that 10x has discontinued the link trees for patent reasons. I won't mention the company that is sort of shutting them down on this activity, but it's a real shame because it's actually a very good protocol, both in the laboratory and in the analysis. Okay, thanks a lot, Tyler. We will move on. It's a great pleasure to introduce the next speaker, who is Roderick Tigo from the CRG. He's the unit head, the program head for bioinformatics at the CRG, and he's going to talk to us about efficient and accurate methods for gene annotation. Good, Roderick, can you unmute yourself? You've got your picture and share your screen, please.
Can you hear me and see my presentation?
Yeah. Yes.
Okay, thank you for inviting me here. So as we have just heard, that's the output of the genome projects. That is the genome sequence of the organisms that we are studying. And the sequence is of relative value unless we are able to map where the genes are in this, this sequence. And I actually will talk about this problem, which I think is quite relevant, and it's not maybe a little bit under studied at this point. So the first mapping of first mapping of genes into a chromosome is more than 100 years ago. Was the work of Sturtevant and Morgan, based on the hypothesis that the distance between two genes in a chromosome was proportional to the frequency with which the characters encoded by the genes were inherited together. This was quite amazing because at that time in 1913, nothing was known about the nature of the hereditary material. It actually took many years, the experiments of Avery, McLeod, McCarthy, Hershey, and Chase, to show that DNA was the hereditary material, the equivalence between genes and proteins, with an atom, the discovery of the DNA structure and the sequencing of the first protein, and the discovery of the mRNA, the ciphering of the genetic code in the early 60s, up to the understanding of how the flow of the information travels from the DNA sequence to the protein sequences. Actually, this is the central dogma of molecular biology, and actually is, in a sense, our understanding of finally of the molecular nature of the gene. So the genes are, in a sense, the regions of the genome that are transcribed to RNA and eventually post-processed to be exported into the cytosol in eukaryotic organisms and then translated into proteins. So this actually offered a way of identifying the genes into the genomic sequences, which is actually sequencing the genomes and then sequencing the RNAs and mapping the RNAs into the genome. That would produce actually a map of the gene, a gene annotation of the genome of interest. Of course, this was of very little practical application in the 1970s, because there was no way of sequencing DNA or RNA. So these methods only became available early at the mid of 75, the work of Sanger, Maxim, Gilbert, and others. And it was not until 77 that the first genome of an organism was sequenced, the virus Fix 174. This actually, the number of DNA sequences was growing fast at the second half of the 70s, and early in the 80s, DNA databases were created that store the few hundred DNA sequences that were available at the time. And this was already incredibly important, because just looking at the sequences that had been stored in the databases at the time, which were, as I said, a few hundred, Fickett discovered that the regions that code for proteins had a statistical composition that could be used to distinguish them from non-coding regions. And actually, using the same sequences, Steve Mount, also around the same time, found that around the splice sites, right, the regions that separate exons from introns in eukaryotic genes, exhibit a particular pattern of nucleotide compositions that could be captured by the position weight matrices that the Stormo had just introduced more or less contemporarily. Again, this was very interesting, but was of little practical importance, because there was not many sequences that were obtained, genomic sequences that were obtained at that time. And in fact, the first human genetic map that was published at the end of the 80s was actually published using the method invented by Morgan and Sturtevant, right? This was about but in this case, rather than studying the segregation of phenotypes that could be observed, physical phenotypes, they actually were looking at the random fragment length polymorphisms, RFLPs. These are sequences in the genome that the length of which varies between individuals, and that can be determined without the need of sequencing the DNA. It was not until the Human Genome Project started in the 90s that the interest for developing methods that could identify in an efficient way the genes and the genomic sequences were invented, and mostly they combine this bias in the composition of the sequence and the information that's on the splice signals to identify the coding exons that constitute the genes. There were many. There has been actually, mostly at the time, we're talking in the decade of the 90s, was a huge effort in this area with many different programs invented to predict genes in genomic sequences. As sequences were stored into databases, other methods that were not based on the analysis of the genomic sequence by itself, but also on the comparison of the genomic sequences with other known sequences, were becoming very powerful. In particular, the comparison of genomic sequences with databases of protein sequences was very relevant, because if you find a region in your genome that is similar to the sequence of a known protein, this indicates that the region codes for a protein which is homologous to this one. And not only a comparison with known protein sequences, but the comparison between genome sequences, even though the gene content of the sequences was unknown, could help to identify where the regions were conserved, because regions that code for proteins are more conserved in the genome than regions that are not conserved. And this is the comparison of two genomic regions in the synthetic regions between human and mouse, and the lines in the diagonal indicate the regions that can be aligned. As you can see, the regions that can be aligned are perfectly conserved in the coding exons, are not so well conserved in the introns. If we move to further phylogenetic distance, the conservation is essentially restricted to the protein coding regions. And if you move even further away, this is the comparison of the human with the fish. We see that there is still there is essentially no conservation in genomic regions, but the conservation starts to vanish also in the protein coding regions. So methods were invented to fine-tune phylogenetic distance and comparative genomics to predict where the genes were located. But maybe even more important than this actually was the advances in capacity of sequencing RNA, not directly, as you know, but by copying into cDNAs. Methods were invented that could clone simultaneously thousands of RNAs from the cell, thanks to the fact that all processed RNAs share the same sequence at the 3' end. And then methods were actually invented for taking these clone sequences and very quickly sequence the 5' and the 3' end of this, and these were called express sequence tags, and were very important into all the efforts to identify the genes encoded in genomic sequences. And actually, in the practice to identify the genes, and I will not go into the details of this because that's essentially recapitulating what I have said. So essentially, methods that are used to predict genes in genomic sequences make use of all this information from information intrinsic to the genomic sequence, as I said, protein coding bias, splice signals, stop signals, etc., to similarity to other protein sequences, similarity, intergenome similarities, mapping of cDNAs into the genome, etc., etc., etc. And for instance, there are many programs now that are based into the combination of all this information. Maybe the most, maybe the best known example is Ensemble pipeline at the EVI. Still, how well these methods work, that was something that we were interested in the first decade of the 2000s, and we organized this ENCODE within the framework of ENCODE, this Genome Annotation Assessment Project. And essentially, the conclusion is that computational methods, as is written here in the abstract, the multiple transcript accuracy, taking into account alternative splicing, reached only approximately 40 to 50% accuracy. So this actually led to the realization that computational methods alone, not even using evidence coming from RNA sequencing, was sufficient to produce a high quality annotation of the human genome. And the GenCode consortium was created in 2003, first as part of the ENCODE project, and since then has been working into creating the reference annotation for human and for mouse. Essentially, I will again not go into details, but there are three main components into the gene code pipeline. There is the computational predictions, the experimental validation of these computational predictions, and then the manual annotation, right? It's the third component, the manual annotation, which is carried out by the Havana team that at some point had more than 20 people looking specifically at the annotations to decide which were reliable and which were not. And gene code, together with RefSeq in CBI, the United States, are probably the two now. Gene code ensemble, right? Gene code and ensemble for human are the same thing, or essentially the same thing. Have the two reference annotations for the human genome sequence. Of course, in the mid-2000s, there was a huge technological advance with the invention of the instruments like parallel sequencing, and RNA-seq in particular was becoming the tool of election to characterize the transcriptomes of cells, tissues, organisms, conditions, etc. As you know, this is a difficult problem because in the mid-2000s, most of the dominant technology was Illumina that produced very, very short reads, and the problem of transcript reconstruction is actually a very difficult problem. And actually, there was based on, I mean, as you know, RNA-seq has really contributed a lot to change our understanding of the transcriptional activity of the genomes, with essentially the understanding that a large fraction of the genome it's expressed with that long non-coding RNAs rival or surpass a number of protein coding genes, and there is a huge amount of alternative splicing. Still for gene annotation, really short read RNA-seq has limited value, and in this sort of evaluation done in the framework of ENCODE, it's called ERGAS, the conclusion was that assembly of complete isoform structures poses a major challenge even though all constituent elements are identified. So still from short read sequences, it's very difficult to get to full length transcripts and good annotation of transcript sequences. Of course, very recently, let's say in the last 5 years or so, there has been a game changer really, which is the long read. We have already heard the long read sequencing, PacBio and Nanopore. And this has already, as I said, within gene code in particular, this is really changing the way in which annotation is being produced. We actually developed a method to annotate long non-coding RNAs that are lowly abundant by first capturing them and then sequencing with long reads, which has contributed to the annotation of the annotation of long non-coding RNAs in gene code. And of course, there is another challenge, ERGAS, which is being actually running at this particular moment, in which data from different samples and sample types from different species, different library protocols, and different sequencing platforms, including mostly, of course, predominantly long read sequencing is now, as I said, this competition, if you want, or this challenge is now being already it's being run, is active, and I believe that by after the summer there will be an evaluation and some sort of publication of the results. Anyway, all these technologies, this capacity of surveying the transcriptional activity of the human genome, in a sense, the capacity of sequencing the genome and the RNA, has changed the view that we had of how the information is encoded into the human genome. That's a typical view of the information encoded into the human genome, let's say 20 years ago, when ENCODE started. This idea of the bits on stream, right? The genes were very well defined. Genes were protein coding genes, very well defined, separated one from the other by large, occasionally intergenic regions. We know that this is a very, very simplified version of how the information is encoded into the human genome. This is the same region the human genome that I was showing before, but as you can see, genes have multiple transcripts. In addition of protein coding genes, there are long non-coding RNAs. Transcripts overlap extensively. There are pseudogenes in the antisense direction very often, etc., etc. And this is just the result of, as you know, surveying only a small fraction of the cellular diversity in the human body. There are now methods that are able to produce full-length isoforms of RNAs in single cells, and these methods that are starting to be published are discovering thousands and thousands of previously unannotated isoforms. So what I have to, of course, the problem that we are facing now is that we are now on the making of this moonshot project, the Earth BioGenome Project, that aims to sequence 1.5 million, the genomes of 1.5 million of the 1.5 million known eukaryotic species that habit on Earth during the next 10 years. Maybe it's now 8 years already. And if you imagine, right, we have GenCode has been working for 20 years, and we haven't really had yet, and we don't have yet a complete catalog of the genes encoded into the human genome. So the task of annotating all the genes that are on Earth, that is the genes of all the species on Earth, is in a sense an impossible task. Even if we use pipelines at Ensemble, so Ensemble takes weeks or months to annotate the genome, this will mean that it will take hundreds of thousands of years to annotate the 1.5 million eukaryotic genomes using current methods for gene annotation. So this is a problem that I think we need to be aware. And I just wanted to, of course, ideally, what you would like is to, because through single cell long read RNA sequencing is the way in which we will be able to characterize all the genes that exist to some extent in a given organism. You could think that the Earth BioGenome Project should be accompanied by cell atlas project. You know, the human cell atlas is the same, that aims to sequence, to characterize by sequencing the RNA of all cells in the human body, right, 10 to the 13. So the idea would be, of course, if you want to have a good annotation of all the genes in all eukaryotic species, you probably have to expand the human cell atlas to 1.5 million species. That's impossible in the short term, although probably it will happen sometime in the future. But I actually want you to look at this plot with some detail and forget the V's here. These are not very relevant here, just the others. So this is the number of genes that have been, that's the evolution of the gene count in GenCode. And as you can see, mostly after the RNA-seq, there has been a huge increase in the number of genes on the GenCode. But if you look at the blue bars, which are the protein coding genes, there hasn't really been a change since GenCode started 20 years ago, right? The number of protein coding genes has remained stable. There hasn't been all these technologies, right? All this capacity of sequencing hasn't really contributed very much to improving or at least to increase the number of protein coding genes that are annotated in the human genome. Actually, if anything has happened, we have less protein coding genes now than we had in 2003. It's not that we have lost them. I mean, we have less annotated protein coding genes now than in 2003. And that's relevant because of the following, right? So it is now becoming very clear through many different words that even though there is this huge transcriptional complexity, this is not so this is the slide that I wanted to show. Even though there is this huge transcriptional complexity, that happens is that across many tissues, it's always the same isoform, the one that's the most highly expressed. Actually, the work of Mark Gonzalez-Porta and Abby Ratner group show that almost 80% of all the mRNA from protein coding genes is always captured by the same isoform. And this work with Michael Tress, Alfonso Valencia shows that this is wrong. What I mean is that most human genes have a single main protein isoform. So what about if rather than trying to do something impossible, which is predict all the genes and transcripts into the eukaryotic genomes, we focus on predicting only one isoform per protein coding genes. And why just one isoform per protein coding genes? Because there are methods that are much more efficient. If you want to predict only one isoform per protein coding genes, there are methods that are very, very efficient. But how reliable are these methods? Is this something that will produce any biologically meaningful information? Here I am evaluating against ensemble, right, in 20 different vertebrate species across the entire vertebrate phylogenetic spectrum of different genome lengths. We're evaluating actually the work of Reliably and Francisco Cámara. We're evaluating 4 gene prediction methods that are still used more or less into this ab initio pipeline. So GeneAD is the program that we actually developed early in 2000. And these are several measures of accuracy. I don't want to go into details, but look into this. This is what fraction of the nucleotides in protein coding genes we predict correctly, and what fraction of the nucleotides that we predict as protein coding are protein coding. And as well known in the field, Augustus is the program that works the best, the most accurate. But GeneAD is actually ranks quite comparable, I would say. The other 2 programs don't have similar accuracy. And they actually have the numbers here of sensitivity and specificity. They don't maybe mean very much, but maybe we can present the numbers in a different way. So what is the fraction of genes for which I predict the protein sequence with at least a given accuracy? So what I can say is that, first, for almost 90% of all the genes annotated in vertebrate genomes, we have a prediction, just an ab initio computational prediction that actually covers this. And for 90% of the genes, at least 20% of the sequence is predicted correctly. So this may actually be enough for functional inferences, and actually not only for that, for then focus on more detail onto where the genes are into the human genome. And is this really important? Well, it's important for one reason, right? It's because of the execution time, right? As I said, ensemble takes weeks or months to annotate the genome. Now, these are what these programs take to annotate the genomes. Augustus actually is very slow. It's very accurate, but the accuracy has a price. GeneAD is very fast. It's extremely fast. It takes 2 seconds to annotate an average megabase per genome. So we can annotate the human genome in an hour. Well, Augustus requires 5 days. And you may say, well, this is of course using I think maybe just 1 core or 2 cores, or I don't know. And you may say, well, of course you can scale this. You have now very powerful computers, and you can just put 100,000 processors in common, and you can speed up.
Roderick, can you please wrap up?
Yes, I'm at the end. But there is an issue, which is the carbon footprint that computation produces. And I think that we should be aware of this, right? That bioinformatics pipeline are maybe actually very, very expensive in terms of carbon footprint. And this actually and this is my last slide. And this is actually a comparison of the carbon footprint of these two programs, right? And you can see that there's a huge difference on the carbon footprint produced running one program or the other. So the conclusions is that fully annotating the 1.5 million eukaryotic species in a Herculean task that will certainly not accomplish in my lifetime, but the high quality protein coding annotation is actually feasible within the time frame of the Earth BioGenome Project. And importantly, protein coding genes capture a large fraction of the biology encoded into the genome sequence. And this is the acknowledgements. This is the work, what small work represented at the end is the work of Ferriol Calvet, mostly with the help of Francisco Cámara. Sorry if I took over the time. Thanks for your attention.
Okay. Okay. Thank you, Roderick.
Okay.
Very nice. We have one question in the Q&A, and the question is, circular RNAs are not currently mapped. Am I wrong? And this is by Romy Peña.
Well, I'm not sure what you mean by circular RNAs are not mapped. Many of the actually RNAs that are mapped into the genome are likely to be circular, right? And they are just mapped as linear. So I think that if they end up into standard RNA-seq libraries, they will be mapped into the human genome, right? And I guess that people could linearize them before mapping.
Okay. I have another question for you. Didn't mention whether RNA modifications might be of any importance in the whole game.
I have no answer to this question, actually. I don't know. Really, for the prediction of protein coding genes, I don't think that they will have a consequence for the prediction. Of course, I didn't mention other classes of genes that are important, like small RNAs. And probably in the case of small RNAs, RNA modifications may have an impact. I don't know what would be the impact in terms of gene annotation, if any.
Okay, good. Thanks a lot. Thanks a lot, Roderick. Roderick, you have a couple of questions that are in the Q&A. Could you please answer them typing?
Yeah, I will. Thank you.
Okay, it's my great pleasure to introduce Per Arrows from the ERTA, just across the mountain there. We've been working together for many years, so looking forward to what you have to tell us.
Thanks, Ivo, for the introduction and for the invitation to. Can you see my presentation?
Yes, but it's not in full screen.
Okay.
Yes, now it's in full screen.
Okay, do you see the pointer?
Yes.
Great. So I'm going to talk about peach and almond and prunus. And prunus includes several species that are worth mentioning because they are important crops: almonds, peaches, apricots, plums, and cherries. And I'm going to talk mainly about almonds and peaches. And these two species that are obviously very different, at least that the product is very different, the peach that is the fruit and the almond that is the seed, but they share many things in common. One is diploid and small genome, and the main thing is that they can be intercrossed and they produce fertile progeny. Actually, this was known for a long time ago, and Charles Darwin thought that peaches actually came from, peaches and almonds were the same species, and that peaches were a breeding product of almonds, which is not the case. One of the important differences of peaches and almonds is that almonds are self-incompatible and then are a cross-pollinating species, whereas almonds don't have the self-incompatibility system at work, and they are mainly selfers. And this has consequences, specifically in terms of variability. So look at it in the way you want. Peaches are have a much lower level of variability than almonds. Several years ago, with Tyler and the CNAG, the Tyler Aliotto and the CNAG, we decided to go ahead and start sequencing the almond genome. We did it mainly with Illumina sequencing and with a little bit of Oxford Nanopore. And then we constructed, we assembled a sequence that covered well the genome of a species, around 1/4 of a gigabase, so a very small sequence. And we found around 27,000 genes, which was more or less what we expected. It was a bit difficult for reasons that Tyler mentioned before. It's that the variety we chose, Texas, is a variety that is highly heterozygous. And then that adds complexity to the analysis. Then we compared the almond and the peach genomes. There is a sequence of Prunus persica, of peaches, that was one of the first in the Rosaceae family, and was one of the last to be done with Sanger sequencing. And it's a high quality genome, really, that it's still the reference for most of the genomes that are used now. And then we compared these two sequences, and we found essentially that they were identical in most respects. Obviously, there were important differences in variability, but the organization of this variability, where the genes were, where the transposable elements were, was almost identical. The one difference that we found that I think that it's interesting is that the transposons, particularly retrotransposons, had more recent insertions in the pea genome than in the almond genome. Saying that there was more movement of transposons in peaches, recent movement than in almonds, in which somehow this could sort of compensate with the loss of variability that implied the self-pollination system in peaches. Transposable elements were also important in other respects, and actually they were essential in the determination of one of the main characters of almond, that is the bitter versus sweet almond taste. This is the key domestication character, and we found that the expression of an enzyme that is in the biosynthesis of amygdalin, that is the principle that produces the bitter taste, was very low when certain insertions of mites were produced in the upstream region of this particular gene. So, well, one thing that we are currently doing is to upgrading this Texas sequence, and we have added some PacBio sequences, and we have clearly improved contiguity, and now we have a sharper tool for genetic analysis. There is also another sequence of almond, the sequence of Lorraine, French variety, that is also based on PacBio, and that is also a help for genetic analysis. Looking at the Prunus genus, in addition to peach and almond, that both are in this Amygdalus subgenus, there are 5 more species that are cultivated: apricots and plums, European and Japanese plums, and then cherries. And they are in different subgenera. And one interesting thing is that if the crosses that you do within a certain subgenus, they are usually compatible. But if you cross the subgenus barrier, this becomes a little bit more difficult. It is possible between Amygdalus and Prunus to a certain extent, or in certain cases, but it is not possible between cherries and the rest that are completely isolated. In Prunus, we say there are more than 200 species, most of them within these 3 subgenera, and all of them are one way or another compatible with cultivated species. So there is here an enormous gene pool that can be used for breeding, obviously, but also that allows to find an enormous amount of natural variability interesting for any purpose. So this genus has attracted attention for different research groups, and now there are 25 genomes de novo sequenced in this genus. Some of them in the cultivated species, and some species with several genomes that have been already sequenced, and others that are from other crops or from the crop wild relatives. I think that there is time already for an analysis at the pangenome level, not only within each species, which is something that is a logical thing to do, but also at the level of genus. And I think that this is possible and reasonable for several reasons. One of the reasons is that the genome of Prunus is very similar in all the cultivated species, at least. There are no major genome rearrangements at all. Of course, there are minor inversions or duplications, but so far we have never found any major rearrangement. So it's small, and by small meaning also that it's relatively cheap compared to other genomes to analyze. And I think that it could be a good model to study evolution. Evolution as the one that occurs within the process of domestication, because you have a very broad transect of intercompatible species that belong more or less to the same subgenus, and also in inter subgenus, and also because of this gradient of intercompatibility that goes from no compatibility at all to full compatibility, I think that this would be also a good model to study domestication. Sorry, to study speciation and reproductive isolation mechanisms. Okay, now I'm trying to move to the next picture unsuccessfully. Well, it seems that there is a problem here. Okay. So this is the map of Prunus, and here you have the positions of Mendelian genes that have been mapped here. There are 56. There are probably more. Well, probably sure there are more. And if you look at them, most of them have an orange or yellow background, meaning that they come from pitch. So 80% of the genes that have been placed on this map are found in the least variable of all the species of Prunus, which is in certain ways is a paradox. One of the things, looking at in more detail this, there are 19 of these genes for which we know the sequence, that they have been cloned, or there are some very clear candidates.
Sorry, but the slides are not moving.
Sorry, they are not moving?
No.
No? Okay. I have a problem, and I don't know which problem I have. Okay, I'll restart again.
Maybe try on sharing the screen and then sharing it again.
Yes, that's what I'm going to do. Do you see it? Well,
we see it, yes,
anyway, I'm going to finish. Can you see this?
Yes.
Okay, good. So, well, the question is that most of the genes for which we know the sequence are either produced, the variability, the morphological variability is produced either by transposable elements, small chromosomal rearrangements, or tandemly repeated genes. And tandemly repeated genes are a very common picture that generates a lot of variability. So structural variability has an enormous role in the variability that we see at the agronomic level. And the way to study this is having very good sequences from the beginning. So these long-read sequences, no matter if it's the whole genome or for specific regions, are going to be crucial for us to understand how this works and how these changes are produced in these regions that produce so much of the of the variability that we see in the phenotype for the future. And now before closing, I just would like to say that thinking of this EBP and the new call that is going on, there are many rosaceous crops in our flora, and many of them also are wild crop relatives to some of the cultivated species like Rosa or Fragaria vesca or Prunus spinosa. So this, I think, that would be perhaps, even that we are not going to be able to sequence everything, at least to start with the things that seem to have more possible applications. Thank you very much for your attention.
Okay, thank you very much, Pera. Nobody, oh, now someone just put up a question. Okay. So the question is by John Bonds, and he asks, have you found any genes related to Xylella resistance?
Well, that would be fantastic, but not yet, at least. It's an important invasive pest now and a center, but to my knowledge, we don't have anything that is resistance or that is known to be resistant, unless still information about the inheritance of the resistance. But we will have to do it.
I have another question for you. Do you, now that you have these reference genomes, do you actually use the reference genomes to look at the variability across different, you know, say different peach trees? Also, do you use this?
Yes, no, that's one of the obvious outcomes, and there are very excellent papers on that that have been produced in peach. Mainly, because peach has been studied with much more depth than any other species. Now we are trying to do this in Almond too.
Mm-hmm. Okay, thanks ever so much, Pera. There might be other questions coming up later in the thing, so I would ask you to then try to answer them in the chat, just to direct you.
Thank you.
Thanks a lot, Pera. So we'll move on to the next presentation, which is by José Antonio Godoy. He is from the Departamento de Ecología Integrativa in the Estación Biológica de Doñana, part of the CSIC. So, José Antonio, great pleasure to have you here, and I can see you're sharing your screen already. If you could put it to full screen, then we might already be there.
Okay, thanks, Ivo. Okay, now everything's
perfect.
Good.
Yes.
So first, I appreciate the invitation to present you our latest research on the links genomics. So to start, just to mention that this is a long story. It started on 2011, and in fact, a progress report was already presented in the first CNAG meeting Symposium back in 2013. And here is, you may recognize a few of the people that became involved early in this project, and it will help you to appreciate what 9 CNAG Symposium, 8 years, and 1 pandemic can do to human bodies. So yes, to acknowledge all the full set of people that contributed to that clearly collective effort. Here are all 5 authors of the main paper, and that was really useful, and we learned a lot along the way. A lot of aspects of genome structure and evolution were researched by different groups and different collaborators. But just to recall and put the emphasis on the ones that are more relevant from what I'm going to talk today, is these 3. First, the origin of the species, the relationship between the Iberian and the Eurasian links. We found with this first genome that the divergence was pretty recent, and especially it was followed by continuous gene flow probably until the present time. We also were able to reconstruct the demographic history, and Here you can see the reconstruction of the 1 Eurasian genome and the 10 Iberian genomes. So you see that they both fluctuating, but Iberian links maintain very small population sizes during most of its history. And we also detected evidence for more recent bottlenecks. Apparently there have been serial bottlenecks in history. So the last one around 300 years ago, and that's probably ended up with the almost extinction of the species by the end of the 20th century. And one of the main things that we learned in that project is that that has clearly impacted the genome of the species, with the Iberian links Martinez here, being the species with the lowest genome-wide diversity reported so far by then, and probably still is, or at least it is among the lowest. And we also found large inbreeding, especially in Doñana population, as revealed by these long runs of homozygosity. Okay, so with this antecedents, what we have been trying to do since this first project was to use those resources and information that was gathered in that early project to deepen our knowledge of what happened to endangered species when they decline and become isolated, what happened to their genomes in more detail. So for that, we have the opportunity to use also the Eurasian links. As I told you, it's a closely related species, and that also offers a suite of demographic situations. We will focus on one population here in Kirov in Russia that's healthy, that's been connected until the present, and in contrast with these other, Norway and Poland, that has peripheral populations that have been isolated and has suffered some genetic erosion. And we are also using the Iberian links, Doñana, the one that was isolated, we estimated like 200 years ago, and have remained at small population sizes in them. With contrast that with Andújar population that was only a few years ago, it was part of a large population and well-connected population. So we decided to compare genetic diversity across the genome for pairs of population, being one bottleneck and one non-bottleneck population in the case of Iberian links, and two bottleneck populations against the one non-bottleneck population of Kirov. And we wanted to go beyond the average genomic diversity. I mean, that was reflected in this slide. So here you can see the intensity and duration of the bottlenecks has resulted in 50%, about 50% diversity loss, average diversity loss in Doñana, 74 in Poland, in Norway, sorry, and 80% in Poland. So we wanted, as I said, we wanted to go beyond average and rather focus on the variance against the variance of the outcome of the bottlenecks across the genome. So we compare diversity between bottleneck and non-bottleneck populations in different chromosomal regions, and I'm showing here in different annotation features that are ranked here according to expected selection intensity, with intergenic representing neutral sequences in one end and ultra-conserved non-coding elements at the other extreme. And here what we represent is average diversity loss. So basically it's the difference between bottleneck and non-bottleneck, and it's a relative difference. So we divide by the sum of the two diversities. So you see different bottlenecks have different average losses, but they differ broadly between features. So the result was, striking result was to see a lower loss or even diversity gains in features that are most selected, like CDS and ultra-conserved elements. And we interpret this to reveal the relaxation of purifying selection that occurs in small and declining populations. We also had the opportunity to try to estimate somehow and characterize the genetic load. I mean, the accumulation of deleterious variation. We know this is a an important component of genetic erosion are occurring in endangered species. But we have two different outcomes possible. I mean, on one side, we have that deleterious variants are accumulating because of the relaxation of purifying selection, as I showed before. But on the other hand, there is a chance in small populations to get rid of most severe deleterious alleles, especially those that are inherited as recessive. So here what we are is following the number of derived alleles in different categories, the neutral intergenic categories. And here it shows CDS. And we are classifying CDS variants into missense tolerated, missense deleterious, and loss of function. And what you see here is relative to the Eurasian links, Iberian links present equal load of neutral or tolerated variants, but a deficit of deleterious variants, either missense or loss of function. And I think this shows the effect of purging, of selection against these deleterious recessives being possible in small populations due to inbreeding. So, and the difference probably has to do more with long-term effective population size differences between these two species rather than with recent bottlenecks. So we wanted also to use the resources as broadly as possible, so included Eurasian links as a study species. We managed to cover a good part of the distribution range, the broad distribution range of these species, and first analyze whole genome-wide neutral data to reconstruct the history. And we defined three main evolutionary units, I would say, or lineages within the species, one Western, Eastern, and then the Caucasus region as another possible refugia. And that was neutral, and we are now trying to approach whether there is adaptive variation segregating among populations. So we did perform genome scans, two kinds of genome scans, of layer FSD approaches and genotype environment association analysis, and identify a few regions with some signal of positive selection or divergent selection within the species. And here is what you get when you use neutral to ordinate the different samples in different populations. So you see the pattern I showed with the East-West and the Caucasus being a little bit divergent. And then here, using only variants that have signals of adaptation, you see a similar picture. I mean, there's still three, but on one hand, all southern population seems to approach, to have similar adaptive variation in these variants. And on the other hand, eastern population that were quite homogeneous neutrally for neutral variation start to separate here, probably because they are responding to different environmental variables. And finally, the last thing we are trying to approach is to expanding even broader to all four lynx species in the lynx genus. This offers a nice scenario because it covers both American and Eurasian species, northern and southern species that eventually overlap the ranges and a number of ecological traits that are different between them. So we thought it would be nice to explore more in detail the divergence and mixture and selection occurring between lynx species. And for that, we also gather a nice set of samples that expand the distribution also of the American species, the bobcat and the Canada lynx, using resequencing data, basically. And one of the issues that I wanted to address with this data set is, well, the history of divergences within the genus. So these are two conflicting genealogies, cladograms representing the relationships of the four species. And as you see, Pardinus is often depicted as a sister species to Eurasian lynx. They are both Eurasian and they share a number of derived traits. And this is what you get with a wide molecular data set. But previous to that, some morphology or even some other molecular data sets were suggesting a different history, where Pardinus is a long diverged species, and Canada lynx and Eurasian lynx are the most recently diverged sister species. So I thought genomes could solve this, but our collaborators already proved this to be the case when they applied phylogenomics, so different windows across the genome, and they were able to infer both types of genealogy, with this one being supported in regions of low recombination. So they interpreted this to mean that are low recombination regions are usually admixture free. So they interpreted this to mean that Pardinus as lynx have admixed extensively in the past, so that even the average of the genome may be reflecting the wrong divergence sequence in the genus, because a great part of the genome in Iberian or Eurasian are in fact shared by admixture. So we wanted to explore this in collaboration with Oscar Lau here at CNAG, also by using complex models that incorporate population size differences, divergence times, and admixtures between species, and even incorporating ghost populations in a context of ABC models enhanced with deep learning. So this is also work in progress. And to end up, I'm not sure how we'll go. Okay, I'll finish up with, well, we thought that in order to progress with this kind of questions, we needed higher quality genomes. So we dedicated some of the resources to generate these novel genomes using third generation sequencing technologies. And here, for example, is the variant links it was assembled based on ONT, Oxford Nanopore sequences, Hi-C, and 10x genome link reads using the strategy Tyler already presented. And here is in comparison to our early genome, the improvement is dramatic in terms of the number of scaffolds that represent the genome, like 100 less in the new version, or the N50, scaffold N50, instead of being 1.6 is 116. So a major improvement in contiguity as well, and important improvements also in gene representation. We are also, data for the Bobcat that they are working on in order to assemble. In this case, data is PacBio and Hi-C only. Preliminary results show that it's certainly a good quality genome, although it still needs some polishing and improvement. And on the other hand, we have Canada lynx that was assembled by our colleagues in Texas A&M. And again, it was also PacBio and Hi-C based. And this one is really close to chromosome level. Okay. I think I'll leave it here. Just a reflection. We heard how this initiative to sequence the whole life in Earth uses an argument that they can help conservation. That's certainly a great idea. We are working on that, but we need to learn first to be able to decide how can genomics best support conservation. And it's not that evident. To start, one part of that is a huge gap between genomics and conservation practice. I mean, there was already a gap between genetics and conservation practice. Genomics just makes things worse in the sense that it's more esoteric or technical and expensive in principle. So yeah, we are collaborating also in initiatives trying to bridge that gap. And just a final acknowledgement to my group, a small group. 3 of them are already doctors. So, and to my collaborators in the aspects I have presented. And thank you all for your attention.
Okay, thanks a lot, Jose Antonio. Okay, good. So you sort of you don't have any questions in the Q&A, but I have something which is similar to what I asked Thomas at the beginning, which is what should the objective be to sort of get animals within the same clade to have as much diversity as possible? Is that the best target, or does it not really matter?
Okay, well, with the variant links we have the question applies to two different scales. So we have two differentiated populations that we showed using ancient and historical DNA that they were recently differentiated. So that makes sense to admixture them, so to mix them, and that's happening already in captivity and also in the wild through translocations. And then we have the issue with the Eurasian links. I mean, they are clearly different species, no question on that, but they have a long history of admixture. The Iberian lynx still is one of the most eroded species on Earth, and although it's recovering, and we made a lot of effort in trying to improve that through the admixture of the populations, it still is. I mean, there's a limit to what you can get. So I don't know. Certainly nobody's proposing to admix intentionally with the Eurasian links, but we showed that happened in the past. It will happen in the future, given the chance. And that certainly probably will introduce genetic variation, although the fitness consequences are not that clear. So we need to be cautious about that.
Okay, thanks a lot, Jose Antonio. Okay, so let's move on. The next presentation is by Paulina Martinez from the University of Santiago de Compostela.
Okay, now is full screen, no?
Yes.
Yeah, okay. So thank you very much to the organizers for inviting me to this very nice meeting. I will tell you the title of my presentation is What Flatfish Genomes Tell Us from the Sea Bottom to the Plate. So we are emphasizing in the title the two lines of research we are working with, both evolutionary biology related to the adaptation of flatfish to the demersal life, and also the importance of flatfish as commercial species, so improving aquaculture. So as a first introduction, I should tell you that flatfishes in the Darwinian times are very controversial because it was difficult to understand how from a bilateral morphology could be obtained a flatfish morphology. So this was a fuel for saltationism, for macromutation. But really, the fossil record demonstrated that intermediate forms with partial migration of ice and progressive flattening was revealed, and really at some time of the evolutionary story, squinting was adaptive for some species. The second important issue related to the flatfish evolution is regarding if they are monophyletic or polyphyletic, to say if they have appeared more than once along the evolutionary history. And the order pleonectiforms are divided into suborders. The so-called modern flatfishes. Sorry, I don't know what has happened.
Yeah, we can't see your screen anymore. What I would suggest is try.
Okay.
Yes, now we can see. Okay, now we can see it again.
So there is a bitter controversy along the last decade between both groups working on phylogeny of flatfish, that some suggested and supported polyphyletic origin and some monophyletic. Really, there are two suborders within the Pleonectiformes order, the modern flatfishes and the suborder Cetoidae, the old flatfishes. And really the support for monophyly was really very, very low. So an image, as you can see, tells more than 1,000 words. This is the typical morphology of a modern flatfish, like Arcturus ocellatus, and the old flatfishes living in the Indian Ocean, very resembling some of the fossil record, so like Cetodesarumei. But in addition to evolutionary issues, we are interested in flatfish breeding programs for improving production, because there are two important species in Spain, but especially the turbot, because it's the most important worldwide flatfish species produced. And the main targets for flatfish, for turbot improving are growth, of course, sex control, because the females grow faster than males, and especially resistance to different parasites and bacteria. Other species that is complementing nowadays the turbot production, and that is increasing very quickly, is the Senegalese sole. The Senegalese sole has a big problem related with the reproduction of males of the F1 in comparison with the wild counterparts that perform much better in aquaculture facilities. But in addition, Senegalese sole has an important problem related to skeleton abnormalities, representing more than 20% of the production. So this is an important issue to be addressed for the future for improving Senegalese sole production. So we started to work with genomics mostly or initially in turbot within the Genome Spain initiative in 2005, and later in the Consolidated Genome Project Aquagenomics, and we released in collaboration with CNAG and other groups in Spain the first whole genome sequence of turbot in 2016 that was published in DNA Research. But later we achieved the 22 macro scaffolds corresponding to the 22 chromosomes of the turbot karyotype using a highly dense genetic map. And finally, 1 month ago, more or less, we have released a final version, a chromosome level version of the turbot genome that includes only 28 scaffolds or contigs assembled using the genetic map that represent more than 95%, these 28 scaffolds, so a very, very huge, important assembly. So with the first version of the turbot genome, we enter in the evolutionary biology issues related with turbot. So we try to understand how this genome was shaped by the adaptation to the demersal life. So we could identify that the genes related to green opsins that are important for low light vision in the sea bottom were expanded in the turbot regarding other flat fishes and regarding the other Acanthopterygii fishes that related with turbot. And in addition, we also could identify several enzymes related with the production of unsaturated fatty acids that were very important for adaptation to the low temperatures where turbot lives. In addition, within the Aquatrace project, we use as reference the turbot genome to look for local adaptation, and the turbot is a very important species for fisheries, but also from the population genomics is very interesting because it is represented across the whole European coast, from the Baltic Sea until the Black Sea. So very important environmental drivers like salinity and temperature are probably shaping the turbot genome. And using DD-RAD sequencing in the turbot genome as reference, we could identify using neutral data set the 4 main units across the European coast, the Baltic Sea, the Atlantic, the Mediterranean, and the Black Sea. And using outlier data set related to divergent and Bayesian selection, we could identify the salinity, the temperature as the main drivers that separated clearly the four main units across Europe. So these two drivers are shaping the turbot genome. We identify candidate genes for these drivers, and we are validating this at the population level. Finally, the genome released in 2016 was used to integrate all previous genomic information related with QTLs for growth, for reproduction, sex determination, and for resistance to different diseases. And using the new Turbo genome, we mine into those genomic regions where the QTLs were located, in blue the growth, in red the reproduction and sex determination, and in green the resistance to different parasites. And we could detect several important candidate genes that were further used for validation at the farm level. All this information are being integrated in the two chips, one highly density SNP chip of 65 Ks, and the other or low density 1.5 Ks. And both are combined to genotype the broodstock and the offspring, and through imputation to improve genomic selection in the target companies. But in 2018, really the long read assembly jump and introduce a new scenario. So the combination long read and short read assembly improve a lot, and a lot of new genomic resources were released in the flat physics. So, for example, we released a new in the Tarbot, but also in the Hollywood, the Brasper, and very recently, 2 weeks ago, it has been released in Nature Genetics, 12 new flat phys genomes. So currently we have 15 flat phys genomes assembled at the chromosome level that are very, very interesting for going ahead, both with evolutionary issues and also with application for improving production. The best issue for sequencing flat phys genomes is that they are very small regarding the Acanthopteri counterparts. For example, Daniurario, you see in the upper part all the flat phys genomes that are between 500 megabases and 700 megabases. And the bilateral Acanthopteri has high genomes, and the Daniurario is 2 or 3 times higher than the flat phys genomes. So because they have a small amount of repetitive DNAs, the assembling using the combination long read polysyn with short read are working very, very well. And the new Turbo genome we released has only 28 scaffolds or contigs covering more than 95% of the Turbo genome. So in this paper of Lu et al., it was very important because they gathered the information of 17 species, including both flat phys and bilaterals closely related, and they demonstrated that the origin of flat phys was polyphyletic. So they emerged at least twice across evolution. That was quite amazing. And a second important issue they presented in this very recent paper is that flat phys evolved very quickly at the end of Cretacic, likely due to a quick adaptive radiation related to the increase of sea level that devised a lot of shallow waters new niches to be covered, and this is the quick evolution that make very difficult to solve the deepened nodes of the flat phys phylogeny. In this paper is also very interesting that no genomic expansions were detected by this group regarding the most important challenges for living in the sea bottom, like hypoxia response, cardiovascular system, visual perception, and immune responses. But a very important genes related with these challenges were detected very quick positive selection for them. And the very interesting issue is that the modern and old flat phys, despite they used some common genes for solving the same challenges, they also use different routes and different genes to respond to the evolutionary demands. So reinforcing the idea that there is a polyphyletic origin. To finish, we are now involved in the FAUN initiative, in particular in the AquaFAUN initiative that is related with functional annotation of aquaculture genomes, the 6 more important fin fish species in the European Union. So the salmon, the rainbow trout, the turbot, the sea bass, the sea bream, and the carp. So we are interested in developing functional genomic atlases using ATAC-seq, ChIP-seq, RNA-seq, and microRNA-seq to develop to obtain early developing map atlas, a body map atlas, and the immunomap atlas that is very important to understand the response to the most important parasites related to this species. And within this project, we apply the new turbo genome and rat-seq sequencing to identify a new gene related to sex determination in turbot. Here there is a GWAS with a zoom from at the chromosome level, at the chromosome 12 level, and at the short region level where narrow identify the SOX2 gene as the main responsible for sex determination. And also at the functional level, we could determine that the SOX gene is highly expressed in females, very close when the gonad begins the proliferation and differentiation between males and females. Now we have 2 new collaborations with Tyler told before regarding the Senegalese soul that we have a very good results. Tyler does not know now this, but we have 98.5% of the Senegalese cell genome assembled in the 21 chromosomes regarding the Senegalese soul stereotype, and we are preparing the material to begin the sequencing of the genome of the grooved carpet cell. So I would like to, I am very pleased to all the group that is working behind these projects, and very appreciate very much the invitation by the organizers. Thank you.
Okay, thank you very much, Paulina. Very nice. I've learned an awful lot about flatfish. I didn't know that anything much about them before. In the Q&A, there's no questions for you, but I think you answered the thing that occurred to me, which was when you said about the FANG project. You said you do different. The idea there is then to do different levels of. Different tissues essentially that go into this thing. But at what level do you go? Do you go into sort of substratifications of different tissues, or you just take a kidney and then the kidney is it, or the liver and the liver is it, or do you then try to dissect the tissue further into different compartments?
No, at this time we are using the whole tissue, okay. But there are many different. For example, at the development level, we are using 12 embryo stages from morula, gastrula, blastula, segmentation, and so on. And within the body map, we are using both juveniles, we are using adults. And finally, in the immunomaps, we are using mostly the head kidney, that is the most important regarding the response to the pathologies. But no, we are not using single cell at this moment. We are preparing single cell in our work, mostly with the olfactive system. Yeah. In turbot and Senegalese souls, because we think it's very important for reproduction and for communication between males and females. So we are preparing single cell bag without touch.
Okay. But I wasn't necessarily referring to single cell. I was just, wondering whether you sort of dissected tissues more into like this part,
Yeah.
Okay, thanks a lot, Paulina. We will move on The next presentation is by Isabel Moody from the Genetics, Microbiology and Statistics Department of the University of Barcelona. So Isabel, can I ask you to share your screen, please?
Yes, I'm on it. So can you see it?
Yes, not yet in full screen. But,
okay, sorry. Good afternoon, everyone, and I'm very sorry for this. And I first want to thank the organizer for inviting me. Today I'm going to explain the Genome project that we did in collaboration with CNAG and how we uncover some adaptations to the aquatic and terrestrial environments in the mayfly. So in the laboratory, we are interested in understanding the genomic basis of evolutionary novelties. And for that, we use the mayfly, Cleondipteron. Here you can see a female individual, an adult, and why mayflies. So first, mayflies are very interesting phylogenetically because they are the sister group to all other winged insects. So they are essential to study, for example, the origin of wings. Also, some species of mayflies have a striking sexual dimorphism. So this is the female, as you saw in the previous slide, and here is a higher magnification of the head, and you see the 2 compound eyes and the 3 ocelli. But when we look at the male, what we observe is that besides the compound eyes and the ocelli, there is an extra set of eyes which are called the turbanate eyes due to this turban shape. And finally, mayflies are also very interested to understanding adaptations to different habitats, as the nymphs or the juveniles are aquatic, whereas the adults are flying insects. So they live on the land or just flying. So we decided that they were interesting enough. So we decided to start working with mayflies, and for that I'm not going to go into the details, but we established a culture in the laboratory, and it was a bit challenging at the beginning, but at the end we managed to do forced copulas, and then we can have females that lay eggs, like hundreds of individuals per offspring, and then we have a lot of individuals every developmental stage, so we can now close the cycle and have a continuous culture. So with that, we decided to, of course, to have also a genome. And for that, we had our inbred line. And we wait for 9 generations. And together with the Synagogue, we did 2 strategies, Illumina and also Nanopore. And we sequenced males. But, and actually, I think the quality, so we have a 98% of Busco, which was quite good. And the genome size is quite small, it's 180 megabases. So, but we not only sequence the genomic DNA, we also sequence multiple transcriptomes from different developmental stages or tissues or organs from nymphs and adults. And why we did that? So first, to help with the gene annotation, but also because we were interested in understanding some gene expression patterns across these different samples. And so this is how the genome looks like. So this is just one small example of our browser. And here is abdominal A, which is a posterior Hox gene. And if we look at our RNA-seq data, we observe, as we expected, that abdominal A is lowly expressed early in embryogenesis, but as development progresses, the expression of this gene increased. But this is something that we already knew. And this is just one example of one gene, and we wanted to do that in a more genome-wide, so using a more genome-wide approach. And for that, we use this type of analysis and fast soft clustering, which give us clusters of genes that are co-expressed in our samples. In this case, these samples that we use here. So, as I said, this is just one example, but we can have multiple clusters with different profiles of gene expression. With that, what we wanted to do then was to focus in the life history traits of mayflies and in this transition between the aquatic and the aerial lifestyles. And for that, we focus especially in clusters with genes with high expression in the nymphal stages or aquatic stages, and the clusters with high expression, clusters with genes with high expression in adults. So we did a gene ontology enrichment to see which type of genes were there. And for the ones highly expressed during the nymphal stages, we found that the terms, the enriched terms were mainly related to sensory perception of chemical stimuli, odor unbinding, etc. Whereas when we look at the terms that were enriched in the clusters with adult genes, the terms that were enriched were mainly related to visual perception. So one thing that we say in the lab is that so the nymphs smell their environment, whereas the adults can see the external stimuli. So we wanted to go a bit more into the details of the chemoperception and the visual perception. And for that, for the chemoperception, it's quite important to take into account that depending on where you are, if you are in the water or you are on the land, so the molecules and how you perceive the molecules is quite different. So the transitions from water to land require physiological, anatomical, and molecular adaptations to the chemosensory systems. And it's also quite important in our case to notice that so from all the chemosensory gene families happening in insects, there are two, the odorant receptor and the odorant binding protein gene families that appear for the first time in insects. So these two gene families have been suggested to be a consequence of the terrestrialization of insects. So we wanted to know what is happening in mayflies. And what we did was to go back to our MFAS clusters, and we checked in which clusters we could find these chemosensory genes. And we found 4 main clusters which contain most of them, especially the odorant binding proteins. And which are these 4 clusters? So those clusters are all clusters that are or nymphal or pre-nymphal, which, so just as a reminder, they are aquatic stages. And not only that, but as we also have many other different organs and tissue transcriptons, we check, for example, where the OBPs were expressed in all our transcriptons. And what we found is that they are mostly expressed in the nymphal heads, but there is a second tissue here that also has a subset of odorant binding proteins highly expressed. And this second tissue are the gills. So the gills are these structures here. There's abdominal gills here that are constantly flapping. And these structures have been always described as respiratory organs or helping the locomotion of mayflies. But in our case, what we are finding here is that they are also a sensory organ, because so when we check some of those odorant binding proteins, the expression within the gill, we found that they are expressed all over the gill plate, and that this expression colocalized with HRP, which is a neural marker. So we describe here a new function or a new role for these organs. So not only, at least not only as a respiratory organ, but also functions as a sensory organ. So this is what we learned from the chemoperception and the aquatic phases, what happens with the visual perception. So the light sensing molecules are the opsins, and in insects there are main 3 opsins: the long wave sensitive opsin, the blue, and the ultraviolet, and the name comes from the type of light they can detect. And in insects, they have undergone different expansions or reductions independently in its lineage. So we looked what happened with Cleondipteron, and not surprisingly, we found also quite expansion, but what was quite interesting was the expansion of the ultraviolet opsins, because with 4 copies is the largest complement described so far in insects. And well, also the blue is not, usually is not duplicated, so that was also quite interesting. And looking a bit the evolutionary scenario of these opsins, these genes, what we found is that not all of them, this expansion, not were all of them Cleon dipteron specific, but some of them were ancestral to the Bettidae family of mayflies. And the most characteristic trait of the Bettidae family of mayflies is the turbanate eye. So at this point, we thought that maybe there was a relationship between the appearance of a new visual system and these new duplicated opsins. And one easy way to test that, as we had transcriptomes from males and females, and if you remember from the beginning, the turbanate eye is only present in males. What we did was to investigate how these opsins were expressed. And we did a differential gene expression between males and females. And what we found is that the two genes that are most upregulated in males, and actually they are not expressed in females at all, were one of the blue opsins and one of the ultraviolet opsins. So at this point, we showed that these genes were male specific because the transcriptomes are from the entire heads, but so are they turbanate eye specific? So we did again in situ hybridizations, and indeed we found that these male specific opsins are expressed in the turbanate eye and not in the other. So, and just to conclude, as I told you at the beginning, mayflies are also essential to study the origin of wings. And we wanted, as we had also the genome and several transcriptomes, we want to tackle also this problem from a transcriptomic point of view. And for that, we use another type of clustering analysis, WGCNA, which generates clusters of co-expressed genes across different tissues. And we did that for mayflies and also for Drosophila and also for Strigamia, but I'm not going into the details. But keep in mind mayflies and Drosophila. So at the end, what we have is a set of modules of clusters with genes that are specifically or sort of specifically expressed in there. So here you can find, for example, the wing module in mayfly, and here the wing disc module in Drosophila. And when we did pairwise comparisons of these clusters, what we found was a high overlap between the genes that are contained in the wing module of mayflies and the genes that are contained in the wing disc module in Drosophila, suggesting a conservation of the wing program, the transcriptomic wing program in the entire pterygote lineage. So with this, I'm going to finish just thanking the Casares lab, especially Fernando, because so this project started at the CABD in Seville, and also the people that helped at the beginning setting up the culture. And as you can imagine, so this is not a one-person effort. So the genome was a collaborative effort, and many people contributed to several aspects of the project, also the funding bodies, and for your attention. Thank you.
Okay, thanks ever so much, Isabel. So there's a couple of there's no questions in the questions yet. Okay, but I have a question for you. So you have nanopore data, right? And you have nanopore data on the nymphs, and you have nanopore data on the adults. Is that correct?
No, the nanopore data is only on the adults.
Okay, because of course an interesting thing to figure out would be, so from the nanopore data, you can pick up DNA modifications. So I don't know which bases are modified in mayflies to do epigenetic regulation, but obviously you could then look at the genes that sort of correlate with what is in the nymphs and what is in the adults, what is on and what is off. Your opinion on that?
Well, so this is our next step. So not looking at exactly DNA modifications, but the plan is to do to do functional genomics and to do ATAC-seq and ChIP-seq probably, but so we have to do it on really cross-reactive antibodies because we don't have, so mayfly is a new model system. But yeah, so the plan is actually, so I think next week we are going to start with a taxic experiment. So the plan is to do that because it's important. So for us, it's important to see the regulation and which are the regulatory regions.
And do you do it on the complete animal, or do you collect it further down?
So the first attacks that we are going to do is embryos, but we are going to, so it's going to be the whole embryo, but it's going to be different stages. But the plan for the midterm is to do a single cell ATAC-seq. So actually, so we already have the single cell RNA-seq samples for the 1218I project, which is a project that we have. So we have already the samples, and hopefully soon will be sequenced. But for the single cell attack, we are also going to do for the 1218I. So we will use the entire head.
Okay. Thanks ever so much, Isabel.
Thank you.
Okay, so I see we have Rosa back. So we've seen a lot of technical problems on the way here with our meeting. Rosa, I think, had a very particular one. So Rosa, I introduce you as the final speaker of the symposium. Rosa is from the eBay over at the CRG building, or the PRBB building, I should say. So yeah, Rosa, please share your screen and you can fire off.
Thank you, Ivo. Let me find the presentation, and I will be ready in a sec.
Okay, that looks good.
Okay, perfect. All right, so let me see if I can see you, because I have two screens and I'm getting a little bit disoriented. Well, thank you all for resisting bravely until the last talk of today's symposium. As Ivo mentioned, I'm Rosa Fernandez. I'm a group leader at the Institute of Evolutionary Biology. And today I will talk about how we can leverage evolutionary genomics to understand animal biodiversity. And the first thing that I would like to do is to introduce the main characters of this story, which are, of course, animals. So when we say the keywords animal biodiversity, this is what most people imagine: beautiful, colorful fish, mammals, big cats, pandas, turtles, and so on. Probably also some iconic invertebrates, spiders, butterflies. But actually, if we were to depict the animal tree of life, the full spectrum of animal biodiversity, it could be something like this. So there's this beautiful poster by Ross Piper that is showing all the different animal phyla. And as you can see, we are familiar with some of them, but probably when you went for a walk in the park, you did not see most of them. So we are familiar with some animal phyla, for example, Craniata, including humans and vertebrates. Some others are also well known, for example, arthropods that include insects and arachnids and scorpions and so on. And we also have some iconic ones, such as mollusks, that includes, for example, mussels and octopi. But however, as you may have realized, some others are weird looking and are small and probably not so common to find in nature as well. For example, we have here cynorrhynchs. They are also called mud dragons. They measure like a few 1 or 2 millimeters, and they live in the sun, between the grains of sand. We have, for example, nematomorphs, also called horsehair worms. They are parasitic worms, and they live inside of other animals such as mollusks and arthropods, or onychophorans, also called velvet worms, and apparently the sister group to arthropods. So if you remember in Thomas' talk this morning, he was mentioning that for the BGP, the main goal at the beginning was to sequence all families, then all genus, then all species. Well, I'm sorry to tell you that we are not there yet, because in fact, for the animal phyla in general, we still don't have representatives in terms of any species sequenced at all for around 1/3 of the animal phyla. So for instance, there are no genome out there of arrowworms, ketognaths, or gnathostomulids, or the mud dragons that I was mentioning, or the velvet worms, or the horsehair worms that I mentioned before. And this is thing kind of shocking in the moment that we are right now with these methodological developments, but we are still at this point in terms of sequencing the main groups or units of biodiversity that could be considered phyla. Actually, if we zoom in some of these phyla, the problem is the same, but at a more shallow systematic level. For example, in this image, we see represented by the outer ring the number of estimated species, and represented by the inner ring the number of known species, described species. We see that the percentage is pretty high in invertebrates. However, it is not the case for most invertebrates. For instance, in insects, apparently, not apparently, but like estimated number of 20% of the species is what's supposed to be described. And 5% for nematodes, 17% for arachnids, and so on and so forth. And of course, many of these super weird lineages that I showed you before are not even represented here. So you can imagine that there's a huge gap in our understanding on biodiversity. And at this point, I would like to make a link to this reflection of Jose Godoy when he was mentioning before that there's a huge gap between biodiversity genomics and conservation. And I would argue that the gap is precisely being filled little by little by evolutionary genomics, because for many of these species that we do not have a lot of information, what we need to do is to start working on some very basic questions. For example, where do they belong in the animal tree of life? Who are they sister to? Or where do they come from in terms of, I don't know, invasive species or endemic species? Where do they live? Or for example, what type of genes do they have? So these are the two different levels that we can work. If we have, of course, a lot of information for other species, and we have very high quality genomes, we can go deep and tackle conservation problems. But in our case, the questions are still very basic, which doesn't mean that they are not interesting, but they are really basic. So this is, again, the field that evolutionary genomics aims to fill in. And my goal today, instead of focusing on a single study, was just to give an overview on some of these basic questions that can be answered by evolutionary genomics in these non-model organisms when we don't know much. The first one, and the most basic one of all of them, would be to resolve the tree of life. As I mentioned before, it is still kind of difficult to believe that we do not know with precision the interrelationships between animal phyla. So this is a little bit outdated, but the truth is that many of the interrelationships are still unresolved. And instead of being represented by a bifurcating tree, we could represent them by a comb, meaning that many different hypotheses have been proposed out there. And of course, this is very limiting, because if we don't know, if we don't have a tree, we cannot do comparative genomics analysis. We cannot polarize the results. So this is a very key question that we need to tackle if we want to move on with more specific tailored questions. And this is just showing animal phyla, but the truth is that if we zoom in and we go the level of family, of genus, there are still many, many unresolved questions in the animal tree of life. And evolutionary genomics definitely have helped improving our knowledge on this sense. A second type of question that we may ask ourselves when we have genomic data for these non-model organisms would be about how their genes have changed, how the gene repertoire, the gene content have changed through times. Which genes are present in some genomes? Are they old genes? Are they new genes? When did they arise? How have they changed? Did they arise through speciation or through duplication? And so on and so forth. The third type of questions that could be easily addressed by genomic data would be about the phylogeographic history of the species. So we know that species move, and we also know that by having genomic information on different populations, we can understand how they have migrated or dispersed or diversify in the space and time. So this is something that that we can tackle with genomic information. And the fourth example of questions, of course there are many others, but the fourth I would like to mention here today, it's about understanding the genomic basis of adaptation. So this is a beautiful figure showing some of the major transitions in insects. The first one is terrestrialization, which is one of the questions that we are most interested in my lab. And we also have evolution of flight, for example. So since Isabel already talked a little bit about this fourth type of question, understanding the genomic basis of adaptation, today I'm going to give you just a few quick examples of studies tackling these three other types of questions: unraveling the tree of life, exploring gene repertoire evolution, and interrogating phylogeographic patterns. So the first story, it's about actually all the animal tree of life. So let me tell you that, so this is a paper that we published a couple of years ago. Let me tell you that the previous attempt to resolve the animal tree of life occurred 11 years before we published this, with only 62 genes or ESTs, express sequence tags. And of course, many interrelationships were still controversial. So what we decided to do is, well, kind of, we wanted to go big, we wanted to represent everything. So we mixed genomes and transcriptomes, because at least we were ready to collect fresh specimens from all animal phyla to do transcriptomes. And of course, we used genomes when they were available. Some of them were better quality, some others were not. But anyway, it was better than that 62 ESTs from 11 years before. And we had represented virtually all animal phyla. And what we did was to create a huge matrix with genomic information. We did a lot of analysis, phylogenomic analysis, which are challenging, particularly with all this huge amount of information. So we really tried to be exhaustive there. And what we found is, on one side, good results, and on the other, not so good results. So regarding the good results, one of the things that we were happy to recover consistently in all the different analyses was the position of these guys here. These are called arrowworms or ketognaths. And we recovered them as sister group to these other three animal phyla. And this was particularly interesting because the position of these guys had been jumping around a lot in the animal tree of life. So finding consistent signal to where they belong, it made us really happy. Another point that we were happy to find was the interrelationships of arthropods, velvet worms, also called onychophorans, and tardigrades. Why is that? So this is the traditional hypothesis: arthropods as the sister group of onychophorans, and then tardigrades as sister group to the other ones. However, many different analyses have recovered arthropods as sister group to nematodes and other nematode-like phyla, such as nematomorphs. And what we recovered with strong support is this traditional view of the sister group of arthropods of onychophorans, which also kind of makes sense from a morphological point of view in terms of segmentation and so on. You have here the velvet worm, and you can see the resemblance from a morphological point of view with arthropods, mediapods, for example, and so on. However, we found many bad results. For one of the main points that was unresolved, that varied depending on the analysis, was the position of ctenophores and sponges. I'm sure most of you have heard about this war, because at this point it's a war between many different teams working on phylogenomics and defending ctenophores or sponges as the earliest splitting lineage in the animal tree of life. Well, in our analysis, half of them pointed to one hypothesis and half of them pointed to the other. So we could not resolve that with precision. And the next bad result that we got concerns the next node, the next most basal node in the animal tree of life, which is the position of cnidarians like jellyfish, corals, and their kind, and placozoans. So we found two competing hypotheses, which were the same ones that other studies have found, supporting that cnidarians could be the sister group of bilaterians and placozoans the sister group to these two, or on the contrary, that placozoans and cnidarians could be the sister group to each other. And the third group of bad results that we found concerns this clade here, which is one of the largest in the animal tree of life. It's called Lophotrochozoa. And this clade encompasses annelids, mollusks, platyhelminths, nemerteans, and some other animals. And what we found is that interrelationships between these animal phyla really change depending on the type of analysis. So we did our best. We mixed different types of information, genomes, transcriptomes with genomes, but genomes. And we really think that once we have more and better genomes, we will be able to resolve some of these controversial phylogenetic relationships. So this is one of the next frontiers, I guess, in animal biodiversity, really tackling the backbone of the animal tree of life. The second example, it's about understanding gene repertoire evolution across animals. So this is a paper that we published last year. And what we had in mind was how did animals reshape their genomes to acquire such a wide variety of forms and functions. Basically, the questions that we had, that we were asking ourselves were, do they have the same type of genes? Are they repurposing old genes for new functions? Are they acquiring new lineage-specific genes? How did the gene repertoire change in the different animal lineages? Is it similar? Can we see similar patterns, or is it specific? So for this, we assembled even a bigger data set than in the previous study, and we included all animal phyla, I think around 240 genomes and transcriptomes favoring genomes, had like more or less half and half. And this is what we did. We did a phylogenomic study to build an atlas of gene repertoire evolution. We wanted to know what happened to each gene in each genome, basically when, how, and how often it was gained, duplicated, or lost in the animal tree of life. And this is what we got. This is a kind of a complex figure, but I would like to draw your attention to three main features. The gray bars represent gene gain, the black bars represent gene loss, and these bubbles, these circles, represent duplication ratios. So the bigger the circles, the more duplications we saw. What are the patterns that we found? What we found is that pretty much all the phyla had similar patterns of gene gain and loss. We found a bimodal pattern of gene gain. We found that most of the genes were already older than the origin of animals. This is where animals originated. These are the outgroups. We have fungi, we have choanoflagellates and their closest relatives. And what we found is that most genes already existed. What we found is that gene gain was relatively low as animals started to diversify into clades. However, the role of gene loss, these black bars, started to gain weight to the point that in some clades, gene loss was really, really high. Of course, gene loss is comparative between clades. So this means that they were getting, as they diversified, they were getting more and more different one clade from the other. What about duplication ratio? Something very interesting, this gene loss that I mentioned, duplication ratio, what we found is that when animals originated, there was a huge duplication. They showed there was a huge duplication of many different genes. So what we hypothesize is that it is not the acquisition of new genes what characterizes animal origins, but the dynamics of these genes. So genes already existed and they duplicated massively at the origin of animals. Then these duplicates probably started to be co-opted to new functions in different lineages, giving them their singularity, and gene loss helped make these clades more different to each other. We checked the function of the, I'm gonna be a little bit brief, I'm not gonna show you more images, but you will need to take my word on that. So we did some, we explored the function of these genes that were duplicated, and we checked that in every single node from the emergence of animals to the tips, to the phylum level. And what we found is that genes that were duplicated in each node, pretty much in every case, had functions, were enriched in functions related to the establishment and development of the neural system. So what we hypothesize is that this is indicating that the neural system has been continuously reshaped throughout evolution, and it has been made independently in the different animal lineages. As I mentioned before, we also found a huge amount of gene loss. And what we hypothesize is that this gene loss probably is related to the duplication. So genes were duplicating and then they were differentially lost in the different lineages. And what we hypothesize is that this could be affecting our phylogenomic inferences, and it's probably why it's so difficult to explain some of these phylogenetic interrelationships. And the third and last example of questions that we may address with evolutionary genomics in this world of non-model organisms when we don't know much, it's about the great phylloxera. So I'm going to show you here one paper that we published last year with the genome of the great phylloxera. So the great phylloxera is this guy here. It's closely related to aphids. As you all know, aphids are pests of plants, and they cause millions of losses in terms of damaging plants and crops. And this one affects very badly the grapevines. They affect the leaves, they affect the stems, they affect the roots. And what we did, we sequenced the genome because we wanted to learn first what kind of genes facilitated its success as a pest. We also wanted to know what it was in the Hemipteran tree of life, because we did not know if they were closely related to aphids or not, or where exactly they were. We wanted to learn about the genes that they had. Were they all genes? Did they have a huge percentage of species-specific genes, etc.? And lastly, we also wanted to learn about the invasion routes, because we knew that the great phylloxera originated in Northern America, and then it conquered Europe. So we wanted to understand a little bit about the origin, combining the genome with population genomics. So regarding the gene content, this is our guy here, Dactylus phyridivitifolia. This is the great phylloxera. And these color bars are representing the number of genes. And the colors, well, starting here to the left, we will have more widely spread genes. And as we move to the right, here in orange, we have genes that are specific of that species or of that lineage. So as we can see here, around 1/3 of the genes resulted to be specific from this species, and compared to other aphids and other closely related insects. So we also noted that many of these genes were highly duplicated. And when we annotated them to have a putative idea of what they were doing, from a biological point of view, we found that many of them were related with nutrition, growth, defense-related processes that may have helped their adaptation to grapes, to the grapevines. And more interestingly, we found that most of them did not show any homology with any databases. Approximately 86% did not yield any hits. So they are very specific genes in this species, and of course they are particularly interesting to start zooming in and finding perhaps targets for insecticides and things like that. Regarding the invasion route taken into Europe, as I mentioned, we knew that it came from Northern America, so we sampled a bunch of population in Northern America, also in Europe, and in other places around the world where the great phylloxera has arrived. South America, South Africa, Asia, and Australia and New Zealand. And we did some population genomics analysis. We explored different scenarios based on the genomic variability found in the different populations. And we found that this pattern was the most supportive one. So what we found is that probably populations from Illinois and Wisconsin conquered Europe, and then most probably a single introduction from France happening to Germany, and then Asia, South Africa, and Australia. So at least now we have a more clear picture about the invasive route of this species. And that's it. Again, my goal here was just to show you some examples of evolutionary genomic studies that we can ask ourselves, questions that we can resolve when we don't have a lot of genomic information, a lot of context. So we will get there, but in the meantime, let's do evolution, which is also very beautiful. And of course, I love this sentence because at the end, it's everything is evolution. Even conservation genomics has to start by this. And that's it. Thank you for staying until the end, and I will be happy to answer any questions.
Okay, thank you, Rosa. You ran over quite dramatically, so
sorry.
I think we're about 20 minutes over the promised end of the meeting. So I think you have one question in the Q&A. I'll let you answer that, and you can maybe do that while I say thank you to everyone. But thanks a lot, very interesting.
Thank you.
So I want to close it here because I know many of you have probably got your stomach, God knows where, want to get something to eat. I want to start out by thanking the speakers. Obviously, very, very interesting presentations. I apologize for the technical problems that we had on the way here. This is obviously always the first few times you do something like this. You have problems that you fight with. I want to thank my co-organizers, Montse and Anna and Rosa. They helped a lot. And then I want to thank the people who supported us here in putting this up, with Katja and Anna and Oscillate, who were managing the Zoom system. I thank you all for participating, and hopefully next time we can do this in person again, and we'll see you then. Look forward to that. Maybe I'm not quite sure if the Zoom system sort of shuts down the moment that we close the session here, and the question that was asked here might get lost at this point. Oh, you answered it. Okay, then I'm going to assume that this matter is sort of sorted. Okay, thanks a lot, everyone, and I wish you a nice rest of your day, and bon appetit. Unfortunately, we can't offer you food this time round, but next time, hopefully again. Okay, thanks a lot.